A catalog of chemical marks on DNA reveals how cells with identical genomes produce distinct identities and functions. This is Planetterrian Daily, episode 156. Every cell carries essentially the same genome, yet executes different programs. The catalog records how chemical modifications on DNA and associated proteins vary by cell type. These marks determine which instructions are read and which remain silent. The resource covers multiple human cell types and quantifies mark distributions at scale. Future releases will add data on how marks shift with age and disease. The effort provides a reference data set for studies of cell identity and dysfunction. The new catalog shows how the same genome produces brain cells, muscle cells, and immune cells through different reading patterns. This resource supplies a baseline for tracking how those patterns shift with age or disease. It sits within the broader effort to understand cell identity as an active, modifiable process rather than a fixed state. The next papers will likely test whether specific mark changes can be used to predict functional decline in tissues. What cell type shift would you most want to see mapped next? A nationally representative sample found that 69% of patients access their test results before their doctors do. A substantial share of those patients report difficulty understanding what the numbers mean. The study highlights gaps in how results are presented and explained. Researchers note that better interfaces could reduce confusion without adding burden to clinical workflows. The work points to opportunities for clearer result summaries and contextual information at the point of access. Follow-on analyses may examine whether certain result formats improve comprehension across demographic groups. Now shifting from the fixed reference map of cell identity, we turn to efforts measuring how those patterns change over time with aging. A new open access commentary outlines how epigenetic clocks and related measures could improve forecasts of disease risk. The piece discusses current performance limits and the need for validation across diverse populations. It emphasizes integration with existing clinical markers rather than replacement. Authors highlight ongoing work to link clock readouts to specific biological mechanisms. Follow-up studies are expected to test clock-guided interventions in prospective cohorts. The commentary appears in Cell Reports Medicine and remains freely available. A few episodes back, it was cellular aging markers. Today's commentary adds how they could integrate with clinical tools. Hotter and drier conditions are lengthening fire seasons and increasing smoke transport. Even distant smoke raises air pollution levels that affect health and productivity. Economist Casey Wichman at Georgia Tech links these exposures to documented economic losses. The analysis separates direct fire damage from indirect effects of particulate matter. Future modeling will incorporate longer seasons into regional cost estimates. The study draws on atmospheric transport data and regional economic records. While clocks track cellular change over years, some bacteria demonstrate rapid, structural adaptations that expand energy capacity in real time. Most cells confine respiratory machinery to the membrane surface. Some bacteria instead build cable-like structures that increase effective surface area for energy generation. This architecture allows greater respiratory capacity without expanding overall cell size. The finding challenges assumptions that membrane area alone limits energy output. Researchers are now mapping the molecular components that assemble these cables. The structures extend into the cytoplasm rather than relying solely on surface folds. Most people picture respiration as a process locked inside the outer membrane of every cell. In reality, certain bacteria build internal cable-like extensions that increase the surface available for energy-producing reactions. Right now, while you listen, microbes in soil and water are using these structures to run more respiratory reactions without enlarging the whole cell. One memorable detail is that this architecture lets them exceed the energy limits once thought to be set by membrane area alone. The mechanism works by physically extending the respiratory machinery into the cytoplasm rather than relying on membrane folds. A practical takeaway is that future synthetic biology designs for efficient microbes may deliberately copy or improve these cables instead of simply adding more membrane. Watch for follow-up studies that identify the exact proteins assembling the cables and test whether similar extensions can be engineered in other organisms. Current global agreements attempt to cover all maritime sustainability issues under single frameworks. New analysis recommends separate rule sets with distinct objectives for different threats. The approach would allow focused mandates on overfishing, habitat protection, and climate impacts. Authors argue one-size-fits-all treaties have produced uneven compliance. Next steps include identifying which existing bodies could host narrower agreements. The recommendation appears in a recent policy-oriented analysis. Recent work connects sleep patterns to protein regulation and broader physiological outcomes. The overview also covers advances in understanding stellar death alongside biological findings. Protein-related mechanisms appear in multiple studies examining cellular maintenance during rest. The compilation underscores how incremental observations accumulate across disciplines. No single study is presented as conclusive on its own. The article groups findings from separate research teams without claiming unified conclusions. A few episodes back, it was sleep's role in cellular maintenance. Today's overview adds connections to protein regulation across multiple studies. Advances in evolutionary data challenge current genus boundaries for humans and close fossil relatives. A Monash University team argues all species from the past 4 to 5 million years belong in Homo. The proposal rests on genetic and morphological continuity that older classifications understate. The study appears in the American Journal of Biological Anthropology. Debate is expected on how the change would affect museum and textbook nomenclature. The authors call for updated naming conventions based on recent fossil and genomic evidence. NTU Singapore researchers surveyed reef sites across the region. 76% lack the growth rates needed to match projected rise under high-emission scenarios. Slower growth threatens both coastal protection and marine habitats. The study combines field measurements with emission pathway modeling. Additional work will examine local factors that might allow some reefs to persist longer. The findings were published after analysis of growth data from multiple reef systems. National data show fewer children interacted with the U.S. child welfare system in the years immediately preceding COVID-19. The downward trend continued through the early pandemic period. The study does not identify specific policy drivers behind the reduction. Authors note the pattern holds across multiple data sources. Ongoing surveillance will track whether rates stabilize or rebound. The analysis draws on administrative records collected before and after 2020. From microbial energy strategies, we move to how sleep influences protein regulation in larger organisms. Long-clawed shrews undergo seasonal skull reshaping that enlarges the snout. Brain volume decreases at the same time, representing an extreme cold weather adaptation. The changes appear reversible when conditions improve. Researchers documented the shifts through repeated imaging of the same animals. The work illustrates how small mammals adjust organ priorities seasonally. The findings come from field and laboratory observations of the species. Seasonal physiological adjustments in small mammals offer another window into adaptive priorities. A nonprofit advocating plant-based diets filed suit against HHS and USDA. The complaint alleges a secret panel altered the food pyramid without standard transparency steps. Plaintiffs argue the process bypassed required scientific review procedures. The case centers on documentation and public input requirements for guideline updates. Court filings detail specific departures from prior advisory committee practices. The suit seeks to restore standard review steps for future revisions. New measurements of cellular and environmental systems continue to refine how we understand energy use, adaptation, and risk. Keep an eye on follow-up work identifying the proteins that build bacterial energy cables. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at JAMA Network. Links in the show notes. Quick tip from the network. Try Fascinating Frontiers next. The most fascinating news from space and science. Every JAMA Network show is free at jamanetwork.com. And on the website, every episode's visuals live in our free image gallery at jamanetwork.com gallery. Royalty-free under Creative Commons. Download with attribution. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.